good morning and good evening to our guests from the US, Europe, Australia, China, and around the world. I'm Rui-Ping Xiao, Deputy Editor of NEJM Frontiers in Medicine. So in spite of, or maybe because of the severe travel restrictions, we are able to meet again virtually for AIMS 2022. More benefits, less harm. So in the last decade, medical AI is finally transitioning from development to deployment. Before we open the dialogue today, let me introduce Dr. Rui-Ping Xiao. So Dr. Xiao certainly wears many hats, and she is a highly accomplished scientist in cardiovascular and metabolic diseases, an inspiring educator and editor, and I'm sure she received many flowers from her students in the last several days on the Teacher's Day, because it's the Teacher's Day in China. So Ping, please.
Thank you, Rui-Ping. That's very kind of you. Dear distinguished guests, my NEJM colleagues and online audience, after successful launch last August, today let me welcome all of you to 2022 annual meeting of AIMS. Medical AI in practice, more benefits, less harm. Rui-Ping just introduced me. I have multiple hats. So I have been an associate editor for the journal for many years, almost 8 years now. Also the managing editor for the Chinese platform, NEJM 医学前沿, and for the new college, a dean of the College of Future Technology at Peking University. As you know, the NEJM has a more than 200-year history. NEJM 医学前沿 is a Chinese platform, a collaboration between NEJM Group and JMAT, launched 6 years ago. The College of Future Technology is exactly 2 years old by this month. Doesn't matter the history, for all the different institutions and bodies, improving healthcare in China and globally is our common vision and our common goal. As the aging society puts increasing strain on the healthcare system, which is exaggerated by the non-even distribution of medical resources around the world. So I strongly believe artificial intelligence can bring radical innovations to mitigate this problem, to benefit people around the world. But AI must be reliable, safe, and ethic at the first place. It also must be improve the efficiency and the accuracy of the expensive clinical trials. Those are the major topics we are going to talk about today. By one estimate, medical AI research and development could add more than 25 billion US dollar to GDP in China in 3 specific areas, namely fast drug discovery, clinical trial optimization, and clinical decision support. Clearly, to reach the full potential of medical AI, we need to team up different bodies, including physicians, scientists, and entrepreneurs. I'm very happy and grateful today we have those leading figures join us. They came from the global AI community. In its 200 years history, the NEJM has advocated and witnessed the development and application of many cutting-edge technologies. Medical AI is no doubt the next frontier of us, for Chinese, for American, for Europeans, for people around the world. Indeed, the cutting-edge medical AI technology with groundbreaking advances requires an open platform to discuss timely and transparently. I'm grateful that my colleagues at NEJM team up with GMAT to provide such a platform. So let me welcome my colleague, Dr. Eric Rubin, Editor-in-Chief of NEJM, to deliver the opening remark. Many of you already know Eric. He is a professor at the Department of Immunology and Infectious Disease at Harvard T.H. Xiao School of Public Health. Eric joined the journal as editor-in-chief in September 2019, just before the pandemic. He taking the responsibility to oversee a lot of the details for all the editorial contents and the policies. Dr. Rubin is also an associate physician specializing in the infectious disease at Brigham and Women's Hospital, and is a professor in the Department of Immunology and Infectious Disease at the Public Health, School of Public Health. So I will not go to the details. I just like to point out he serves as a scientific advisory board member for many different communities, societies. So by the way, he also do a lot of basic research and translational research in addition to be a physician. Now, please join me to welcome our chief editor, Dr. Eric Rubin, to deliver the opening remark. Eric, please.
Thanks, Ping, and I'd like to add my welcome to those of John Fei and Ping, to everyone who can join us. You may wonder why a medical journal would be interested in AI, and I think Ping just outlined the critical reasons. After all, we're interested in improving healthcare and improving the delivery of care to people, and of course, the fundamentals of how the research is done to achieve those ends. AI is one of the new methodologies and perhaps will become one of the basic methodologies. So we are very interested in learning about it, in helping advance AI, and being a part of your community. So thank you for joining us. I want to thank the participants. I want to thank the organizers and all of you who are listening. And I hope that we get to hear a lot of exciting work now and in the future, and that our journal will continue to be part of your community.
Thank you, Eric. So our next speaker, who is going to deliver opening remarks, is Dr. Xianqun Fan. So Dr. Fan is a member of the Chinese Academy of Engineering, vice president of Shanghai Jiao Tong University, and the president of the School of Medicine of the same university. He is a distinguished professor in ophthalmology for more than 30 years, mostly focusing on ocular oncology and optical diseases. He actually uses AI in his research a lot. So let's welcome Dr. Xianqun Fan.
Today, I'm very honored to be here to participate in today's meeting, co-organized by NEJM Group and JMAT, to participate in the 2022 EMS annual meeting, and also to have this opportunity to discuss with leading experts from AI and also from healthcare field. so that together we can discuss the application of medical AI in our daily practice, and also to discuss the future development direction of AI. In recent years, we can see AI technology has been making tremendous progress and also is making huge impact on our everyday life. And also in healthcare field, we are now having intelligent AI integrated into our infrastructures. This will change our conventional treatment models and also treatment strategies and technologies. In the ophthalmology field, we've been using AI to determine the glaucoma, optic neuropathy, and also diabetic retinopathy by using the fundus image. And we have published a lot of relevant articles in NEJM. And also these has advanced the AI application in ophthalmology field. And for the Shanghai Jiao Tong University, number 9 hospital, we've been building our multi-modal database. And also by utilizing these database, we have started the orbital disease diagnosis and also treatment AI-related clinical trials. And for Shanghai Jiao Tong University, we've been paying a lot of attention to AI application in medical daily practice. And also, we've been making some progress. For example, in Rui-Ping Hospital, in Professor Ning Guang's team, they have developed the master diabetes clinical management pathway, and for their master, they have integrated the most advanced diabetes management technologies and strategies, as well as the AI technology. After training the AI and machine learning models, the machine learning and AI will be able to mimic the physicians to provide diabetes management and treatment decisions support and also advices for our physicians, so that we'll be able to improve the standardization of diabetes management. And in the AI Research Institute, as well as the Shanghai Artificial Intelligence Association, together we have announced the artificial intelligence white paper. And in the white paper, we have reviewed and summarized the development and integration of AI. And also, we have analyzed the hot topics of AI research in medical field, and also talked about the major challenges and future development directions of AI in healthcare. And we have established the Digital Healthcare Research Institute in Shanghai Jiao Tong University. We hope that by adopting the digital innovation, we'll be able to further promote the integration of AI and advanced technologies into healthcare practice. We hope that we can use the advanced technologies such as AI to promote the intelligence level of healthcare research, and also can accelerate the transformation of digital healthcare, and also to promote the sustainable development of healthcare. Today ends 2022. More benefits, less harm. I believe that this meeting is a very important platform, and also being held at the perfect timing to allow us to have great exchange and communication to promote the more rapid development of intelligent AI development in healthcare field. And finally, I'd like to wish you all the best, and also wish today's meeting a great success. Thank you very much.
So Dr. Ning is a member of Chinese Academy of Engineering, president of Rui-Ping Hospital, affiliated with Shanghai Jiao Tong University's School of Medicine. So Dr. Ning is best known for his research in endocrine and metabolic diseases, but he is also a very enthusiastic medical AI practitioner and promoter, having not only used AI in his research, both basic and clinical research, but also pushed the application of AI technologies in Rui-Ping Hospital. So let's welcome Dr. Ning to deliver the opening remarks.
Hi, it's a great honor to be invited to take part in this workshop. So firstly, I'm sorry because my colleagues told me this is the Chinese workshop, but and also they prepared the Chinese version for me for the opening remark. But I know this is the English the meetings. For this, I have to translated Chinese version into the English. So firstly, it's my great honor to be invited into involve this the very important the workshop to talk about the medical AR and also in practice the benefits and the help for the patients or the other actions. I think this is very, very important. At the present, we face so many the challenge in the world, include the China. Firstly, the increasing of the chronic disease and also the other the disease, the population is also the increasing. For this, the shortage of the medical resource, I think across the world, we all face the shortage of the medical resource. For this, how to get the solution for this the challenge for the whole medical service in the world. Maybe the medical AR may be the very important for us. For the, I think not only in the medical service, but also in the medical practice and also the hospital, the management. As mentioned by the Professor Fan, in Rui-Ping Hospital, also is affiliated to Shanghai Jiaotong University School of Medicine. We made some progress in the medical AR and also included the medical service, just improve the feeling of the patients in our hospital. And second, we also do a lot of the medical AR in the medical practice and improve the efficiency of the doctors to make the practice for the patients. And second, more important, the hospital, the management. And also we do a lot of the medical AR to improve the hospital management, as mentioned by the Dr. Fan. So in the diabetes, so also is more important. We also apply a lot of the medical AR in the management of the diabetes patients. For example, we do some medical AR to assistant for the doctors to make the practice in their service, and also we provide some medical AR to the patients. They can bring this medical AR back to home and help them to control the blood sugars. So all of this we can put it together and we establish the MMC. That means the metabolic disease, the management centers. And this center we establish, and we establish the more than 1,000 the clinics around, across the whole China. So all of these centers, we will take the same the medical AR technology, and also we follow the same the medical standard, and to provide the same the service for the diabetes patients. So this is a practice in China, and also today this is my great honor, and I'm also happy to learn some of the new informations on the medical AR in the medical service. Thank you so much.
Yeah, thank you so much, Dr. Ning, for delivering the opening remarks. And so our next session would be session 1, Intelligent Clinical Trials, Transforming Through AI-Enabled Technology. So as we know, artificial intelligence and machine learning tools in particular are transforming how clinical development occurs, delivering significant time and cost efficiencies, while providing better and faster insights to inform decision-making. So I think it's a very timely session to discuss these topics. Our first speaker is James Zhou from Stanford University. He is an assistant professor at Department of Biomedical Data Science and also a member of the very famous Institute for Human-Centered Artificial Intelligence. So I first got to know him last year when someone recommended a Nature paper he published in April 2021, where he optimized the exclusion-inclusion criteria of some oncology clinical trials. And I'm sure he will talk about it today. So welcome, Dr. Zhou, for the speech.
Hi, everyone. I am James Zhou. I'm a professor at Stanford University, and I'm very excited to share with you our recent works on AI-enabled in silico clinical trials to advance precision medicine. Clinical trials, as you know, is really the gatekeeper and the gold standard, or the bottleneck of all of medicine. Right? So each trial can cost tens of millions of dollars to run. And you could start with hundreds of trials, but only one of them would actually really be successful and make it into patients, right? Which means that the average cost of taking a treatment all the way to the patient can be billions of dollars. Given the impact, right, and the importance of these clinical trials, anything that we can do to make these trials more efficient and better can potentially be extremely useful. So motivated by this, we've been developing approaches to take large-scale real-world data, especially clinical genomics data extracted from the medical records, and to use this data to computationally emulate clinical trials using what we're calling in silico trials. So I'll describe how this works, and as well as 2 applications of using in silico trials to help us to design actual randomized clinical trials to make these trials more efficient and more inclusive. And the second application is on how to use these in silico clinical trials to discover genomic biomarkers, which can then be used to inform treatment recommendation. So in the first part, I'll describe this computational platform called Trial Pathfinder, which is described in this paper that we published in Nature last year. So the goal of Trial Pathfinder is to computationally emulate clinical trials to help us to design the eligibility criteria of actual clinical trials. This work is led by my student, Rishan Liu, and we're quite excited that Trial Pathfinder is now being used by Roche Genentech for informing the designs of some of their new cancer trials. So the problem that we want to solve is in designing clinical trials, right? So how to design clinical trials is very complicated because the trial would have to decide which patients are eligible to participate to enroll in the trial. So if you look at a typical clinical trial, it will have pages and pages of PDF containing rules to exclude patients from participating based on their previous lab values, comorbidities, medical history. And it's being recognized now that many of these clinical trials are overly restrictive, so they're excluding too many patients. And this is a big pain point for patients because many patients would actually like to enroll in clinical trials to get access to the latest treatments, but they're not eligible. It's also a big bottleneck for drug developers, for companies, because a large number of cancer trials and other clinical trials fail to enroll the minimal number of patients. It also means that the so-called patients that participate in the trial are what's so-called Olympic athletes among the patients, and how well the treatments work on those Olympic athletes of patients do not really reflect how well the treatments work on the real-world population. So one question is, how do people currently design clinical trials? The designs of trials need to balance, right, the safety of patients with demonstrating the efficacy of the treatment. And unfortunately, right now, we don't really have good data-driven approaches to help us to design these eligibility criteria of these trials. So to illustrate this, I'm showing you is actually some screenshots that I took of an actual trial that's being developed. Right, so the first thing that the teams usually do is they would copy and paste over some protocol from a previous trial that they had done before. And then they would go through the line by line and see if can they actually modify any of the eligibility rules. Right? So basically each line here corresponds to one eligibility rule, which means it's one of the rules that are used to exclude patients. Right? The first line basically excludes patients that have neutrophil counts that are outside of some range. And you can actually see a discussion in the sidebar of a Google Doc that says that, okay, like basically, can you actually relax this threshold, right, to enroll more patients? But somebody else would say that we don't really have enough evidence to relax the threshold, so let's keep it to be the same as before to be for consistency. You can see similar discussions for other lab values that are used to exclude patients. And the upshot of this is, as you can see, that there's actually a lot of heuristics that are used to decide what eligibility rules and thresholds to use to select patients. And as a consequence, many different trials end up using very different rules for including or excluding patients. Right, so as an illustration of this, here I'm showing you 10 different trials from lung cancer. Each row here corresponds to one of those trials, and each column corresponds to one of the commonly used rules to exclude patients. Right, so they could be based on biomarkers, previous diagnosis, or some range of laboratory test values. And I color-coded a square yellow if that particular trial used that rule to exclude patients, otherwise it's colored gray. So the details are not so important, but the main takeaway here is that it's even visually quite clear that similar trials looking at similar drugs often use quite very different rules to exclude patients. So people are recognizing that this is really quite a huge challenge and huge problem. So for example, in the US, the National Cancer Institute have put out a mandate, right, that the eligibility criteria for all of these cancer trials needs to be simplified and standardized to enroll more patients. But how to do this in a data-driven way is challenging, and this is where we want to help. So the idea here is really a question about counterfactuals, right? So we would like to know, right, among the patients who were excluded from participating in clinical trials, then which of them would actually have safely benefited from the treatment? And our idea is quite intuitive. The idea here is that many of these patients who were previously excluded, they actually end up taking the same treatment after the drug comes on the market, even if they're not eligible for the original trial. So this means that we can answer these counterfactual questions by going to the medical records of these patients and see what actually happened to them, what's their outcome. And to do this, we worked with the Flatiron network of medical records, which covers about 2 million cancer patients across 280 cancer clinics in the US. So it's a very well-curated longitudinal medical records of a large number of patients. And here's the main idea of these in silico trials, right? So we basically use the medical records of these large number of patients to emulate clinical trials in silico. Here's the illustration, right? So let's say you have some particular trial with some treatment, some control, and the set of rules used to exclude patients, right? Then we can go into the medical records and basically identify which of the patients actually took the treatment drug, right, and satisfies this eligibility rules of, for example, bilirubin less than 1. That creates one digital cohort of patients. Then we can also look at which patients actually took a control treatment, which could be chemotherapy, for example. Then that creates a second digital cohort. These two digital cohorts could look very different. They could have different demographics. So then we can computationally and statistically match these two cohorts, right, so that they look very similar. And then after matching, right, then we can compute estimates the effect size of the treatment using things like hazard ratios. Now, the beauty of doing this approach in silico is that we can use on the computer to very easily change the different eligibility rules, right? So for example, instead of having bilirubin less than 1, I can actually enroll more patients by relaxing this threshold from 1 to 2. So this means that now I have larger digital cohorts because there are now more patients that are satisfies this less restrictive condition. I can then similarly match the distributions and then estimate the effects of the treatment. Now, the intuition here is that when I relax the bilirubin, right, from 1 to 2, right, and if I don't see too much changes, right, in the outcome of the patients, in the hazards, their survival or adverse events, then that gives us quite strong data-driven evidence to suggest that we could potentially enroll more patients by using this more relaxed condition. So that's the high-level intuition of the algorithm. So one question that people might have is that, well, how does this work if the drug is actually not on the market, right? Because if it's not on the market, then where do we find the information from the medical records? If the drug is not on the market, then what we do is that we can often look for drugs that have similar mechanisms of action or similar drugs, right? So oftentimes, for example, if you're looking at the new immunotherapy, then we can find a previous immunotherapy that are already on the market that have similar effects. Then we can use the data from that previous drug to do this analysis. The other question that people often have is that, what about confounders, right, in these observational data? So this is a real limitation with any of these analysis using real-world data. In this case, we benefit from having quite highly detailed electronic health records, right, longitudinal information over many years. And we also validate our results using data from actual randomized clinical trials. So then just to summarize, right, so this trial pathfinder approach that we have here, right, it's basically, takes these designs of clinical trials, encode them, right, into mathematical rules, and then we apply those rules, right, to the medical records of these hundreds of cancer clinics to build the digital cohorts of patients. And once we have digital cohorts, then we can estimate outcomes like adverse events and survival outcomes. And now what we do is to basically do millions of these in silico trials in parallel. Each time we do it, we actually use different combinations of eligibility rules, right? So this is useful because then it enables us to test what's the impact of having different eligibility thresholds to enroll fewer or more patients. And at the very end, right, we take all of this information from these millions of simulated clinical trials, and we use a particular approach that we developed called Shapley values, right, to aggregate all of that information into one set of data-driven recommendations for the eligibility design of clinical trials. Right, so here's basically the application of these 2 lung cancer trials that we mentioned before, right? So for each of these lung cancer trials, for each of the rows, right, the output of trial pathfinder is a set of specific recommendations on which of these eligibility rules, inclusion or exclusion criteria, are the most important and most useful to use in each of these trials. And we applied this to several different cancer types, including lung cancer, melanoma, and breast cancer. So here I'm showing you the results from lung cancer. The most important row to look at is basically the last row, which shows the average results. So essentially what we're looking here is that if I just look at the original eligibility criteria that were actually used in these original clinical trials, right, we can enroll a relatively limited number of patients, about 1,500 patients. But if I use the data-driven criteria that's recommended by Trial Pathfinder, on average, we can more than double the number of eligible patients, which is great. And it's also very interesting that the actual hazard ratios of this larger number of patients is even lower than the hazard ratio under the original trial criteria, and lower hazard ratio is actually better. So this suggests that many of the patients who were systematically excluded from participating in clinical trials, they would have actually benefited from the trial's treatments to a similar or even greater extent as the original trial cohorts, as compared to these original Olympic athletes among the patients. And critically, right, by taking this data-driven approach and enrolling more patients, we're actually enabling a lot more women, more minorities, and more older patients to participate, to access clinical trials. So just to summarize this part, right, so Trial Pathfinder uses this idea of AI-enabled in silico trials to help us to design actual clinical trials to make them more inclusive and more efficient, right? And we have validated our results using data from randomized clinical trials, as well as data from different independent cohorts. And while I focus on cancer here, I think these similar ideas can also be extended to other diseases. So we're very excited. This algorithm trial pathfinder was recognized by the pharma industry as one of the top data science innovations of 2021. So we've also taken this idea of in silico trials and used it to go one step further to actually further advance precision medicine, precision oncology. So the goal of precision medicine, right, is to take biomarkers of individual specific patients and use those biomarkers to identify what treatments is really the best suited for that particular patient. So to do this analysis, right, we basically link together the medical records from Flatiron Network with genomic profiling of these tumor patients, right? So we use the Foundation Medicine test, which provides a next generation sequencing based test for over 500 key cancer driver genes. And we basically are able to have a matched data set, a linked data set of over 40,000 cancer patients, right, with their mutation profiles sequenced from these NGS tests. And we also have the treatment and outcome information derived from the medical records. And from this information, right, we're really interested in looking for mutation and drug interactions. So here's an example of such an interaction, right, between a particular gene, APC, which is the tumor suppressor, and immunotherapies. So basically, when we look at patients with bladder cancer, right, some of these patients are given immunotherapies. And what we found is that among those patients who are given immunotherapies, those patients, if these patients had mutations in this APC gene, they have actually had much better survival outcome compared to patients who did not have mutations in this APC gene. Right, so the orange curve here are the patients who had mutations compared to the blue curve of those patients who did not have mutations. And conversely, among the patients, bladder cancer patients with mutations in the APC gene, we also find that the patients that had immunotherapies ends up have better survival outcomes compared to patients who are given other treatments, which targeted therapies or chemotherapies. Right, so these two information combined really suggests that for patients that had APC mutation in bladder cancer, really immunotherapy is really the best treatment for those patients. Right, so this is actually really not known before because APC was not very well studied in relation to immunotherapy in bladder cancer patients, but it's something that we could actually identify from our real world data sets. In this particular case, we're able to support this finding using data from a randomized clinical trial. So basically, Roche had done a clinical trial of 900 plus bladder cancer patients on immunotherapy. They had done similar kinds of genomic profiling of each patient before they started the treatment. And in this randomized trial, we also find that there's a very strong and positive interaction between APC mutations and positive response to immunotherapy, which is shown in this very low hazard ratio, which means that the patients who have APC mutation or given immunotherapy have much better response. And mathematically, these kind of interactions is really captured in this equation where we basically have an interaction term specifically for genotypes, which in this case will be mutations in APC gene, and the treatment, which in this case will be immunotherapies. So that's just one example. And by going through these large medical records systematically, we were able to identify hundreds, over 450 similarly strong mutation treatment interactions. Right? So each of those interactions will correspond to a predictive biomarker that says that the patients with that mutation tends to have better outcome when given a specific treatment. So here in this visualization, I'm just showing you some summaries of a subset of this interactions that we discovered. So they're broken down by different cancer types. Here's lung cancer or colorectal cancer, breast cancer, etc. Within each of the cancer types, we also have different classes of treatments, which could be immunotherapies or chemotherapies or targeted therapies. So I color-coded each of these genes to be red if that gene has a negative interaction with the treatment. Right? So for example, EGFR would have a negative interaction with immunotherapy, which means that in our data sets, right, patients had EGFR that had worse outcome when given immunotherapies. And I colored the gene blue if there's corresponds to a positive interaction, right? So basically, for example, patients with KRAS mutations have better outcomes when given immunotherapies, right? So, and the size of the gene basically is proportional to how many patients would have those mutations, right? So this is very useful for generating new hypotheses, right, for further analysis. And a lot of these mutation treatment interactions can be used to inform treatment recommendation for patients, right, to advance precision medicine. So we have created a website called precisioncancer.org, which visualizes and releases publicly all of this information and findings that we have for many of these markers, right? So if you're interested, then you can look up the particular mutations of interest, and it will tell you what are the treatments that have positive or negative interactions with that mutation. So just to summarize, right, so I hope to show you how we can use this computational idea, right, of in silico clinical trials to help us simulate these really expensive actual clinical trials, right? And these in silico clinical trials, by leveraging these large-scale real-world data, can help us to to design new clinical trials in a data-driven approach to make the actual clinical trials more inclusive and more efficient. And it can also help us to discover new biomarkers that predict treatment outcomes, right? So that can be used to recommend specific treatments to specific patients. And finally, and I think there are many other exciting applications, and here I listed the key references for our works. So I want to acknowledge my students, Rishan Liu, and our close collaborators, Shamra and Ryan from Genentech for this work. So thank you very much.
Thank you so much, Dr. Zhou. And we're going to have time to discuss for an Q&A during panel discussion. So let's move on to our next speaker and actually next speakers. So Alex Zhavoronkov, he is the founder of and CEO of In Silico Medicine, and this is a clinical stage AI-powered global biotechnology company, and they actually have some platform to predict the phase 2 clinical trial outcomes. And he is going to present this talk together with Alex Aliper, who is co-founder and president of In Silico Medicine. And Alex Aliper pioneered the application of AI in multi-omics data for drug discovery and drug repurposing. So let's welcome to Alex to give our presentation. Thank you.
Hello, ladies and gentlemen. My name is Alex Zhavoronkov, and I'm a founder and CEO of In Silico Medicine. And together with me is Alex Aliper, the president of In Silico. Today, we're going to talk about interpretable artificial intelligence for prediction of clinical trials outcomes. And without further ado, I'll start the presentation. So, as I mentioned, the title of the talk today is Interpretable Artificial Intelligence for Prediction of Clinical Trial Outcomes. I will start with introducing the company and our entire framework, and Alex Aliper will feature in Clinico, our new tool for interpretable prediction of clinical trials outcomes. So first of all the legal disclaimers, no forward-looking statements, not an investment advice. And In Silico was founded in 2014, originally in Baltimore, Maryland. And then in 2018, we decided to move to Hong Kong to be right next to massive contract research organizations because we decided to develop our own drugs. Currently, we are about 250 scientists globally. Many of us are AI and machine learning experts, but we also have a lot of drug hunters. And we hire a lot of full-time equivalents within many contract research organizations in China and globally. So as you know, this is the regular framework for traditional drug discovery. It starts with target ID. then you generate hits, small molecule hits or biologics, and then you progress all the way into phase 1 clinical trials in humans, and you go to submission and launch. Usually this process takes at least $2 billion, and excluding target discovery. Target discovery, in my opinion, is the most complex and the most risky and most promising area of drug discovery where artificial intelligence can help a lot. Here on the bottom, you see the probability of success, number of years it takes, and the amount of costs in millions of dollars. So artificial intelligence can help accelerate this process pretty dramatically. So what we decided to do, we of course mapped out all the processes in drug discovery and development that could be accelerated by AI, and transformed ourselves into a company which is an yin-yang of AI and drug discovery, powered by laboratory robotics. So we have the full scale and end-to-end AI capabilities with the teams that are focusing on generative AI for chemistry and biology, also clinical trials prediction team, CADD and AI team. However, we also have a very substantial team taking care of biology, medicinal chemistry, preclinical, CMC, and we also sit on top of many contract research organizations that perform laboratory work for us and generate massive amounts of data. We also have our own lab. So again, we are a platform and a pipeline. So on the platform side, we've set, to my knowledge, several records, went in 18 months from 0 to preclinical candidate, and under 30 months to phase 1. We have published work where we showed that in 46 days, we can go for after a known target with a lot of data, DDR1, all the way to mice with synthesized compounds. We have a lot of papers, many patents, patent applications, and we incorporate over $2 trillion of public data in our engines. In terms of the validated pipeline, we have 31 programs, 29 are fully owned. One is in phase 1 clinical trials right now, and we expect multiple preclinical candidates to be nominated in the very near future. So this was possible by an end-to-end system, which took us over 6 years to build before we started using it internally for drug discovery. It starts with PandaOmics, which predicts the outcomes of which identifies novel targets using many different data types. We have Chemistry 42, system which generates novel molecules with desired properties. And we have InClinico, which predicts the outcomes of clinical trials and is interpretable. That's what we're going to be presenting today. We've used those systems to, as demo, to demonstrate that we can identify a novel target, generate small molecules, predict the outcomes of clinical trials, purpose the target molecule association towards a specific disease, and go all the way into preclinical candidate in under 18 months. So February 24th, 2021, we nominated for our first PCC. Since then, we've nominated 8 in under 16 months. And the same program went into phase 1 clinical trial in under 30 months. Again, we've used all 3 systems, PandaOmics, Chemistry 42, and InClinico, to achieve this result. And currently, to my knowledge, is the first one of its kind. Again, February 24th this year, we've nominated we started phase 1. So since then, we've established a very substantial pipeline of therapeutic assets, starting from target X. We have not disclosed it yet, so it's in phase 1. After completing phase 0, and we have a number of other promising targets, either novel or older targets, established targets going after new indications, and some are made for commercial partnering purposes. So now I'm going to pass the word to Alex Aliper, the president of InSilico, who is in charge of InClinico development. Alex, please take the wheel.
Thank you, Alex. Over the next 10 minutes, I'm going to present to you InClinico. InClinico is a comprehensive framework specifically designed to predict the outcomes of phase 2 clinical trials. Next slide, please. The core utility of InClinico is multi-modal forecast of single phase 1 clinical trials probability of success. So we want to make sure we estimate the success of any given phase 2 clinical trial, which features small molecule has a target profile which with a single target and is also a single drug modality. Additional capabilities of a clinical platform include highlighting and spotting red flags in clinical trial design. So we want to make sure we capture those and raise the red flags. We can recommend additional indications and therapeutic areas for a given therapeutic asset. So purposing and repurposing are in the toolkit of the clinical platform. And we have a detailed interactive reporting that can describe weak and strong points of individual clinical trial and provide suggestions to the investigator. Next slide, please. In summary, the platform and clinical platform leverages multiple layers of data, diverse data types. We have omics data component, structural data component. We also evaluate trial design and trial protocol, and additional clinical and preclinical data is included if available. We also monitor a lot of public domain data and text data, including grants, publications, and patents. In order for us to derive a prediction, we leverage an ensemble of deep neural networks that work together to derive a probability of success value, feature impacts on probabilities. So we're deep black boxing the prediction and making sure it is interpretable and actionable. And we can also derive safety forecast, providing estimates on on-target and off-target toxicity. Next slide, please. On this slide, we outline the framework in terms of different scores we implement in a clinical platform. The first score is a trial design score, which features multiple elements of trial design protocol, which evaluates blinding, target accrual for a given clinical trial, clinical sites and composition of clinical sites, comparator choice, number of arms, and other features in order to evaluate how likely the given trial is going to pass and transform into phase 3 and move forward. The next score is a target choice score, and this score is extremely important to evaluate the mechanistic link between a given target and the disease. So target choice score sits on top of many layers of data, including omics data, clinical level data, protein-protein interactions and signaling pathways, drug-target interactions, and many different data types to see how well given mechanistic hypothesis fits the disease profile. We also have scores linked to patient eligibility criteria, which track comorbidities and pretreatments, and also multiple scores in the admit tox area, which we all use to make a prediction on probability of success. Next slide, please. In terms of data, as I mentioned, we are leveraging vast libraries of data, and in terms of data quantity, we're tracking 1.20 thousand clinical trials which cover 11,000 drugs and over 6,000 indications. Next slide, please. In terms of validation, and validation sits at the core of a predictive algorithm such as in clinical, we have spent significant amount of time ensuring we are carefully validating all modules and models included in the platform. One of the techniques how we validate the tool is called quasi-prospective validation. By quasi-prospective, I mean the training model where we train on data up to a certain time point. Let's say we will train the data on this slide starting from 1995 to 2017. And because we have timestamp data, we can actually use the time period 2018 onwards as a validation data set. So when we're doing it this way, we're ensuring we're preventing any information leak between the training and test set data. And by doing it this way, we were able to correctly predict more than 80% of phase 2 to phase 3 transitions. Next slide. And just to showcase the applicability of the platform to different domains, to different therapeutic areas, on this bar plot, we can see the quasi-prospective performance of a clinical platform in each therapeutic area. Overall, you can see the performance varies, but overall, we're able to get way beyond 0.8 in precision for both failure and success. And we're able to get reliable predictions in most therapeutic areas. Next slide, please. So we did a quasi-prospective validation, but in order for us to truly understand and test the platform, we actually went through a series of prospective validation exercises. This paper you see in this slide, we published in 2020 and evaluated the pipeline of Novartis, the upcoming readouts for a certain subset of trials were expected to come in late 2020, early 2021. On the next slide, we see the table that summarizes the results. So actually in the table, you can see individual clinical trials from Novartis pipeline as rows, and probability, the last column in the table indicates a probability of success to reach phase 3 clinical trial. We can see that since 2020, 4 trials read out. Out of those 4, 1 was predicted to fail, the phase 2 clinical trial of fevipiprant in its application, so childhood onset asthma. We predicted it to fail, the trial in fact failed. The other 3 trials were predicted to succeed, and in fact, they succeeded. So this is just one of the applications where we showed how we can use the platform in triaging and dissecting individual pharmaceutical companies' portfolios. Next slide, please. We went one step further and did a prospective validation on small and mid-cap pharma companies. So we thought that it is paramount to also tap into more innovative programs, which are more prevalent in the smaller biotechs. On this slide, you can see a scatter plot where target choice score is plotted against the trial design score. So those are predicted scores. And the dots are colored based on the actual readout. So you can see that majority of green points, they accumulated in the areas of high target design and target choice score. So the predictions are in fact correct. And based on this result, we see that the precision for success is over 90%. On the other hand, you can see that the red points, the red dots here, they mostly accumulated in the left bottom rectangle, and they contributed to a precision for failure of about 80%. Next slide, please. This year, we published another round of prospective validation predictions. We evaluated 40 ongoing clinical trials, and 19 of those were predicted to succeed, and 21 trials were predicted to fail. We put this paper out early August, and we already have some of the readouts coming through and confirming our prediction. On the next slide, we showcase 2 trials that read out since our publication, since we put it on ResearchGate. And for both trials, so one trial was predicted to succeed, and the other trial was predicted to fail, and the predictions were in fact correct. So the first one covers the Syncore Pharma positive data on phase 2 clinical trial. The second one is Aptinyx phase 2b clinical trial in fibromyalgia, which predicted to fail, and it in fact failed. Next slide. So it is worth mentioning that all those readouts in small and mid-sized biotech companies, they oftentimes cause significant stock price shifts. And we can make our bets based on the clinical trial outcome prediction scores, and can put more confidence into certain therapeutic programs and even in certain companies when you're triaging biotech portfolios. So you can essentially get your own portfolio based on a high confidence prediction from the in-clinical platform. Next slide. It is also a very important feature of a clinical platform that we're aiming to de-black box the prediction, meaning we want to evaluate the impact of each feature in the overall estimate of probability of success. So here on this example, the Aptinix trial I mentioned earlier is evaluated in the context of trial design and protocol features by our platform. And you can see what you can see to the right is a so-called SHAP plot. The SHAP plot dissects individual impact of every feature in the data set and assigns it a score. So basically here you can see that target accrual of 300 individuals contributed positively to the trial design outlook, while our features provided negative contribution into overall design trial design probability of success. This utility of a platform is extremely useful for actionable analysis of clinical trials and even clinical trial planning. Next slide. Now I want to tease a little bit the platform itself. So initially, the platform was conceptualized as a backend solution we can leverage to predict and improve our confidence in our own therapeutic pipeline and assets. But since then, we have made decision to make it clinical into a commercial product. And we've implemented an interface and UI design for in-clinical as a product. And you can see here, this is a sneak peek into the main page of the product, which we yet to release. So stay tuned for that. And in the main page, we show different statistics on several therapeutic areas where we track individual catalysts and clinical trials by company, by therapeutic area, by region. And we provide an extensive outlook into individual clinical trials. And you can actually score your own clinical trials inside the platform with a score your trial feature we have in the system. So stay tuned for the release. It's going to be coming up quite soon. Next slide. This is how individual clinical trial page looks like, where we have individual description of a single trial, therapeutic areas, and importantly, target choice score and trial design score are indicated here. And it is also indicated that those are high confidence predictions, those predictions that we put more weight in. We're also linking press releases and announcements onto individual clinical trial pages, as well as 10K annual reports from publicly traded companies. Next slide, please. So overall, if we summarize applications of a platform, the in-clinical can really help prioritize clinical and preclinical programs and triage those to subset those programs which would have higher chance of succeeding in the clinic. It can also identify red flags in the current or ongoing clinical trials and make corrections before first patient is enrolled. We can also identify what went wrong in the past trials, so do sort of post-mortem analysis of clinical trials and see whether we could have predicted the failure or success. We can also keep great degree of visibility on the competition. So competitive landscape analysis is something this tool can really help with, where you can contrast your own programs with competitor programs or do competitive analysis. So it's all very relevant to commercial application in pharma industry. And from an investor standpoint, we can truly identify companies and therapeutic pipeline projects, which are more likely to be successful and ultimately have more confidence in them going forward. And we can correctly adjust net present value of assets for risk and value companies appropriately with greater returns and lower cost of capital. Overall, InClinical presents a unique product where we combine big data and AI analytics, and we spent over 5 years developing this product. We're not rushing the launch of the platform. We spent quite substantial time on prospective validation because it's truly an indicator of utility and power of the platform. And on the next slide, I would just like to thank everyone for your attention.
Thank you for the presentation. And so next, we are going to move to panel discussion. And so we'll have Dr. Alex Aliper, Dr. Guang Ning, Dr. Rubin, Dr. Xiao, and Dr. James Zhou to participate in this panel discussion. And the discussion will be moderated by Dr. Jeffrey Drazen. Ping, could you introduce Jeff?
Sure, great honor. I think many of you already met Dr. Jeff Drazen. He was our editor-in-chief for a long time, actually 18 years, had been the chief editor for the New England Journal of Medicine. In 2019, he retired from this position, but he still very actively and play an important leadership role in our journal. Recently, he launched a new journal, is NEJM Evidence. Also, the thinking leader for the medical AI 3 years ago, when he had the vision to talk with Charlotte, Jianfei, and me. Actually, our partner, JMAT, colleagues from JMAT, Paula and Fong, we think this is a very important area. The journal cannot afford to miss it. So that's why we have this platform, AIMS. Dr. Drazen have done amazing job when he was the editor-in-chief. The immunotherapy was born in that area, in that period of time when Jeff was in charge of the journal. There are also many very important events, as you remember, during the time for Ebola, for Zika viral epidemics. Dr. Drazen led the team, the editorial team, published a lot of important papers in our journal. I also wish to take this opportunity to thank Jeff for his vision and commitment to promote the east of the world, of the earth. Gary and I became the associate editors of the journal in almost 8 years ago, in 2014. Actually, that was Jeff's idea and endorsed by the whole editorial board. Not only because I was elected, but because it made such a difference for the journal and for the whole community, medical community. Now you can see in China, clinical research is catching up rapidly because this kind of leadership penetrate to our hospitals, to the medical schools, to industry. Jeff, thank you so much for your leadership and for your vision, the ground vision, make such difference. Now let's, Jeff, could you please chair this session?
Thank you, Ping. I'm happy to chair the session, and good morning, good evening, good afternoon to everyone, depending on where in the world you are. So what we've heard in this session from the two speakers was that the world needs a better way to approach the interface between drug discovery and clinical application. Right now, that interface is set in the performance of clinical trials, which are designed to answer specific questions about the performance of a therapeutic or a diagnostic in clinical settings. The idea, which we heard from both of the speakers, was to cut down the cost and speed this up. And since we all share the common goal of trying to improve human health through better diagnostics and therapeutics, it would really make sense. It would be wonderful if we could make these things work. We heard two presentations that suggest that there are certain settings where they do work. That is to say, where we could potentially shortcut some of the time that's spent. But both of these are now at the stages of promissory notes. They've shown that they can predict things that are going to happen. But the issues that I see and that I think is important for us to discuss is this. AI needs to be trained. The training is done on data that are extant. And there are three issues that we really need to think about in those data as we train our models. The first is that there are major ecological changes that can occur. The models that we heard about today were trained during the period of immunotherapy in cancer. But just as an example, suppose that sometime in the next year, there's a major revolution in cancer therapy, one that we didn't think of. We didn't think of the immunotherapy advances until they occurred. How do we deal with large changes, I would say epochal changes in the way we think about a disease based on the outcomes of clinical trials? Remember, clinical trials are really very expensive scientific experiments, which when they work, teach us something about the biology of disease in addition to being helpful for patients. So that's the first thing, is that we have to be concerned that by using old data to predict the future, the biases that exist in that old data are going to be brought forward if we use these to make decisions about not bringing forward things because the old data suggested they wouldn't work. So it makes it difficult for really innovative ideas to come forward. That's the first issue. The second issue that I see is that the data that we have, even if there hasn't been a change in the ecology of care, is biased in many ways. If we work with patients' data from clinical trials, we know that there's bias in the people who are in clinical trials. And so therefore, it may not be all that useful. Now, what Professor Xiao is doing is to try to see how this plays out by looking at the data from real-world experience. But remember, that real-world experience is colored by the physicians prescribing based on the data that they had already. So they said, my patient doesn't exactly fit this, what was in the trial, but I think it was good enough. And what he's saying is maybe good enough is enough, but the patients who didn't get the drug prescribed, aren't going to be in that database. And so it's only a marginal look forward. It may be enough to make a difference. The third is take the patient's perspective, especially patients with diseases that are fatal or potentially fatal. You know, we think about in a lot of the examples that we heard about from Xiao or cancer examples, where the disease generally, if left untreated, can be very severe or fatal. But there are many diseases which are nuisances. I spend a lot of time with asthma. Asthma is very low mortality, but has a huge impact on quality of life. And in that kind of setting, would a patient want to be excluded or included when there may or may not be all that much of an advantage over what's available? Finally, we need to think about adverse events. With cancer, patients put up with a lot of adverse events because they know if they don't, they may die. But in other conditions, adverse events can be the rate-limiting step. A drug that may work, but makes you sick in the process isn't going to get very far. Now, we know that AI is smart enough to be able to handle all these things, but we need to sort of think about the biases that come when we use old data to help us think about the future. So how do we move forward where we're not so driven by what had happened in the past in a way to get to the future? Now, we heard from James and Alex, AZ, and AA, Alex A and Alex Z, and James. So I'd like to hear now from Ping in first, and then Dr. Rubin second, about how you feel about these issues. Will AI help us advance this particular phase of drug discovery in a way that helps what we want to happen, which is to speed the process, lower the cost, but most importantly, make it something that's fair and at the same time doesn't squelch innovation? So first Ping, and then Eric, I'd like you to give me your opinion about these issues as they reflect on this, and then we'll hear from our two speakers how they respond to what you say. Ping?
Ping, you're on mute.
Thank you, Jeff, for the nice summary. Also the question, the important question. This is extremely important for developing countries, for those imbalanced area, for remote area. The medical care is not really as developed as in US or Europe. For example, in China, we have such a big population for the oncology patients and non-communicable disease patients. When we develop clinical trials, it's so expensive. It's not really affordable for our ordinary people, you know. They cannot really have the opportunity. So this kind of AI platform could really make things more economical, more reliable, and safer for the subjects, for the patients, and for the medical care system. How to do that is definitely a very rapidly developing area. Our speakers already showed us, based on different database, based on those kind of real world data or the RCT data, we can have different picture. But still, there's so much to learn, so much to develop. I think this kind of discussion will speed up the mature of this whole field. The technology will benefit, not only Chinese, but people around the world. I'd like to hear more from the speakers, also from Dr. Rubin. Thank you.
And actually, I would like to hear both from Dr. Rubin and Dr. Ning. But first, Dr. Rubin and then Dr. Ning, you will, we'd like to get your opinion about where this is leading before we sort of get a rebuttal, I imagine, from Alex and James. So, Eric?
Well, I share your concerns, Jeff, but I will point out a couple of things. First, on the issue of how well predictions will apply to radically new therapies, I think that is an issue, and I would bet that the algorithms that Insilico uses show much more reliability for a new drug of an existing class than they do for a drug with a radically new target or a radically new mechanism. That has to be the case. We know that the fifth H2 blocker is likely to look like the fourth H2 blocker, for example, in decreasing gastric acid secretion. Whereas something that is wildly new, the breakthrough kinds of drugs, are going to be harder to predict because we just don't have the same sort of database that we do for other drugs. The pharmaceutical industry is a relatively risk-averse industry. They have to be in part because phase 3 trials are so expensive. And I am concerned that signals of risk may prompt companies to not move forward with breakthrough kinds of drugs. And I will be interested in hearing a response to that. On one of your other points, Jeff, I also think that you're right that the patients who are taking drugs in the real world were prescribed those drugs in part because of the resemblance to the patients who are in the trial. And therefore, it isn't a random or far from a random collection of people. And the controls are going to systematically be different from the patients included in a real world sample. But let me counter that with the fact that I don't think that the system of doing randomized controlled trials for every question is sustainable. They're fantastically expensive. As James pointed out, they are not, they're very specific for a population, and it's very difficult to generalize from them. So there are certainly going to have to be compromises made in how we both develop new drugs and how we apply the use of those drugs to different populations that weren't captured in the original randomized controlled trials. And I am very optimistic that AI can help us with that, even though, like the original RCTs on which they were based, they will be imperfect. I'll stop there.
Thank you, Eric. Dr. Ning, and I will point out that we need to finish in 15 minutes. So Dr. Ning, you have 4 or 5 minutes, and then that'll give us 3 or 4 minutes for James and Alex to speak before I summarize. Go ahead, Dr. Ning.
Hi, Jeff. I think this is a very important and useful speech. So I just have 3 concerns. Firstly, so I don't know what's the difference in the ethnic population. So maybe the model, the AR model, just developed in United States. However, we can use apply or not apply in the Chinese populations. This is the first concern. The second, the different disease. So the first speech just focus on the cancer. So if we want to apply this AR model in the metabolic disease, but the quality difference between the two diseases. So for the cancer, we know a lot of the target genes, but for the metabolic disease, maybe the environment factor plays the more important role. So how to develop the AR model in the metabolic disease is the second concern. So third, so I think that this is very useful, the AR model, but how to make the application in China. So you know, in China we have so many the new the pharmaco. So they like to develop the new drugs. Then maybe we can collaborate with the two speakers and make the applications in China. I finish.
All right. Thank you, Dr. Ning. All right, so I'm going to ask Alex to go first and James, because he went first before, to go last. We have about 12 minutes. So
Okay.
Alex, you have 4 minutes or so. I'll cut you off when you get there, but go ahead for now.
Thanks, Jeff. And thanks everybody for, really relevant questions. I'm going to start with your points, Jeff, first. So first point about the training and bias introduced in the training, because we're training on a past experience. So it's actually important to note that we're using different training, like sliding windows, and covering different time periods in the training process. So one example I showed in the presentation is a training window up to 2016, but we have many more training windows where we train on up-to-date data. 2021 is the latest one. And we actually cover different time periods, so we can essentially capture those modern developments and model developments and modalities which are introduced recently. So that's first point. Second, it is true that we also need to be mindful of the fact that we only can predict what can be predicted. So we are not making extraordinary claims that we can predict everything. We're focused on first of all small molecules which are targeted, clear MOA, and we also don't do combinations are more challenging to predict. So we're doing single agent as a first tier of predictions. And yeah, it's really important to only predict those events, those clinical trials that can be predicted and supported by data. Third point on a bias. So bias will always exist, and our philosophy here is just to carefully track the data and transparently account for bias. We know that different clinical sites, they have different, productivity historically. Different geographies have different, factors working for them. Placebo effect in CNS indications in US has to be accounted for, and so on and so forth. So we just have to transparently take it into account and forecast based on that. So lastly, forecasting is only one component of predictive platform we're building. We also believe that it can be useful for planning purposes, so we can essentially better plan the clinical trial outline and execute it in a more efficient way. So we think that this platform can be applicable there as well. And in terms of some other points, we're not restricting ourselves to the US. US-based clinical trials. We're global. We're assessing global clinical trial markets, and we focus on not only on oncology but on various modalities of diseases and therapeutic areas. And we showed our performance actually varies depending on the nature of a disease and therapeutic area. So we actually try to be therapeutic area conscious and narrow down to those therapeutic areas where we can actually show performance. And yeah, finally, we also believe that China and being able to predict clinical trial outcomes in China is really important. And we're actually our front-runner program in IPF. So the molecule that was designed by AI. So we found the target, some mechanistic insights derived from AI, and small molecule itself generated by our generative chemistry engine is currently in phase 1 in China. And we believe that our platform will also help to augment and guide the clinical development process to, well, elevate the likelihood of success.
Thank you. James?
Great, thank you. First, I want to thank Jeff and Ping and Ning and Eric for the very thoughtful discussions and very interesting questions. So I agree with Jeff and with the other commentators that first, that there are limitations of the existing AI algorithms that we should be aware of, especially related to limitations in terms of biased data and limitations in terms of the ability of AI algorithms to extrapolate to really new drugs, to entirely new drugs. And many of these limitations are because these AI algorithms are still in early stages of development, especially in relation to clinical trials. So that being said, I think it's also important to think about what are the alternatives and whether the AI algorithms, even with its limitations, provide some complementary insights beyond the current state of practice, right? Because we don't want perfect to be the enemy of some advance and progress. So first, related to the data, right, the data biases. So I would say that the alternative is that we know that the clinical trial population is often very narrow. It's often like these Olympic athletes among the patients. The real world data still contains biases, but in many cases, we think that it's relatively more diverse compared to the randomized clinical trial populations. And especially in cancer cases, we do find also many instances of patients even being prescribed treatments off guideline or off label, right? So essentially, as we're able to look at broader populations through the medical records, also across the other countries, I think that's going to provide us some interesting complementary information about how to, beyond the very narrow clinical trial cohort. Now, the real world data still have some biases. But here I also believe that's like many of the existing ways that people are designing the clinical trials, eligibility criteria in particular, are themselves also quite biased, right? Because oftentimes people think about the traditional notions that there are normal ranges for different lab values and conditions for designing the eligibility criteria of clinical trials. And the so-called normal ranges are often based on what people did before many years ago with very narrow slices of the population, right? Often based on white males in the US, right? So I think by looking at more diverse populations in the EHR, at least we can make some progress in taking a data-driven approach to broadening those eligibility criteria.
Thank you. We're running out of time, so I appreciate that. I was going to ask Eric for a brief summary before I wrap up. Eric?
So very briefly, Jeff, I think we were just discussing some of the limitations inherent in AI using historical data, which is of necessity. I just want to stress the opportunity here, though, which is that I don't think that medical research, which is slow clinical research, which is slow and extraordinarily expensive, is a sustainable way of producing new drugs, new therapeutics, and new ways of delivering care. And I think that this emphasis on using AI as a tool for medical research rather than just a tool for interpreting EKGs is a really important area. And I'm really very much appreciate all the effort that people are putting into it.
Thank you, Eric. And I actually agree. I think there are 3 areas here that we need to think about for the future. One is genomics. This was mentioned actually by both Alex and James. And I think that if we had a way of selecting people who would benefit, and in cancer, we have both the genetics of the person and the genomics of their tumor. So it gets to be a slightly more complicated problem. But I think that if we're going to move forward, we want to be able to find more pure cultures, as it were. That is to say, to find people who we think are likely to do better. And I think that what James has shown us is that we can find those people based on prior data, and then whether we can find them through EHRs and genomic data to help look in the future, we might be able to streamline the clinical trial process. So that, I think, is an important advance. I think the second area, which is kind of really important, is the forecasting of adverse events and the mitigation of those adverse events. Why do people have them? Is there a way to understand that? And there are a few examples now of adverse events where we know they're going to happen, and we can eliminate them. And I think one of the things that drug companies, one of the reasons that they're so conservative is that a drug can be really good therapeutically, but its adverse events can kill it, especially if its target indication isn't going to kill the patient or isn't going to make the patient very sick. So I think that we want to advance. I agree that the way we're doing things now is slow and expensive. But as we move forward, it's important always to keep in mind what's going to be best for our patients. How can we move things forward in a way that will advance therapeutics? And as Ping has pointed out, especially in countries where they don't have the resources and infrastructure that may be otherwise available in other places. We need to be able to do this so that the whole world can benefit. The example that we've seen from COVID, where we have very effective vaccines that have been deployed that were actually a medical miracle, but yet they haven't been able to be deployed around the world for economic reasons. So but we need to think how we can make drugs more affordable, treatments more affordable, diagnostics more affordable, and to use AI in the process as a tool. I think that we need to use it like any other tool. I'm a very avid woodworker. I work with tools all the time. They can make things that are beautiful. Every tool you use carries risk. And as long as we're cognizant of the risk, let's use the tools to create something beautiful. And that beautiful thing can be a therapeutic or a diagnostic that will advance human health. They'll help health professionals around the world do their job better, more quickly, at lower cost. So we have hope for AI. We've seen a couple of examples. We're looking for more. So for those of you out there listening, think about how we can use AI to improve the discovery, the testing, and then the broad deployment of new diagnostics and therapeutics, because that's what this is all about. Jean-Fei, back to you.
Thank you, Jeff. Thank you so much for this very insightful comments to close this session. And thank you so much for our guests. Whenever you are and whenever the time it is, thank you so much. So I think this closes our first session. Thank you. We just had a very successful session 1 to discuss, the clinical trials, how AI can help clinical trials and how clinical trials can move forward AI. And for wider deployment in healthcare, AI has to be trusted. And a couple of high-profile failures of AI in clinical practice due to data shift or other reasons is a prime example of this trustworthiness, the importance of this. But what are AI scientists, the clinicians, and the biomedical scientists doing to ensure the AI safety? We'll have some top experts from a very diverse background to discuss this topic in session 2. In AI We Trust, the level of evidence that ensures AI robustness. So our first speaker is Dr. Kang Zhang. He is chair professor and vice dean for research and director of Center for Biomedicine and Innovations in Macau University of Science and Technology. So he obtains his MD and PhD program in it's a combination program in Harvard Medical School and MIT, and he got his PhD in genetics from Harvard University. And he actually is he is a practicing clinician. He actually finished emergency eye surgery yesterday, and so we're glad he can he has the energy to deliver the talk today. But he is also an expert in many different areas, such as genetics, epigenetics, stem cells, nanoengineering, and 3D printing, and of course, artificial intelligence. So without further ado, let's welcome Dr. Kang Zhang to give a presentation. So, Dr. Zhang.
Thank you very much, Jeff, for your introduction. Let me share my slides. So I want to talk about AI-physician interactions for better healthcare delivery. I list here in the first slide some of the challenges in applying AI in healthcare and biomedicine. Those challenges are, first of all, data challenges. So the data, as all you know, are very highly often in different modalities, difficult to combine. They're unlabeled, unstructured, so it's not very easy to use. There are also technological challenges, such as integration of immense clinical and high throughput biological data, high multitask learning on disease diagnosis, therapy, and outcome predictions. And obviously, the cost-effective computing capacities. In this talk, I want to touch upon three areas. First is AI assistance. In this way, in a broad stroke, making foundation models or pre-trained models, in this case, transfer learning for specific healthcare tasks. I want to talk about AI empowerment, where the AI can create new knowledge and diagnostic tools, such as a well-known case of AlphaFold 2. I'll give you some of the examples of ours. Lastly, physician AI interactions and trust, in which how the AI can help physician making decisions and how we can address AI black box, such as using a occlusion test. As we know, one of the challenges is actually rapid growth and explosion of electromedical records. Another one is diversity and complexity of digital health data. The third one is uneven distribution of healthcare resources. So to address those issues and make a good implementation of AI technology, we wrote an article a few years ago. In this article, at the healthcare space, AI is poised to play a major role across the spectrum of the healthcare domains, including diagnostics, therapeutics, population health management, administration and regulations, and policymaking. And with the regard of the vast quantity, and accessibility of electromedical records, those will influence decision-making processes in multiple fronts, including for patients, physicians, healthcare systems, healthcare providers, and regulatory bodies. I want to give first a few examples of how AI can assist in making pre-trained models, introducing some of the well-known technologies into the healthcare space to empower us to do the diagnostic test, and which one of the benefits is significant reduction of required label data. Also, physician AI interactions, in which we address the issue of black box with the occlusion test. So like Jeff said, I'm an ophthalmologist. I want to use some examples of diagnosis of eye disease using transfer learning and deep learning. In this case, we're looking at some very common retinal diseases called diabetic retinopathy and macular degeneration. The macula is the center of the retina, which is subserving the central vision and color vision. So in this study, we were able to actually deploy an artificial intelligence system using transfer learning and effectively classify images for macular degeneration and diabetic retinopathy. And using transfer learning, we're able to, again, learn and to differentiate bacterial pneumonia versus viral pneumonia on chest x-rays. And this has a broad generalizable applications in biomedical imaging. So one of the challenges of making a very good diagnosis in computer is actually using AI. Is this very, very characteristic features of big data and small tests. In those cases, AI performs very, very well. However, not all the texts or any areas will have a huge amount of label data or good data to start building the model. So we actually came up with a solution using the existing data, in this case, ImageNet, a very well-known general picture collections of data set, for which it has cars and apples or human or cats. We first train the CNN to recognize images. Then we use this pre-trained model or framework and shift them or transfer them to diagnose the common retinal diseases. In this way, CMV or wet AMD, diabetic macular edema, early macular degeneration, such as Drazen or differentiating the normal retinas. So we used this tool called optical coherence tomography, which is very commonly used, the most commonly used tools in ophthalmology. We used about 100,000 images, label images, and trained the model, and then validated them in about 1,000 images. And then we carried out the occlusion test. The AI performed comparable to a retinal specialist in US by this amount of data, which is about creating about 99% AUC and a 95% accuracy. However, I would like to say that giving enough data, the AI should be able to almost obtain 100% accuracy for diagnosis type of disease. In order to address the end-to-end prediction, therefore a black box phenomenon in AI, we come out with a simple solution called an occlusion test. In this test, what we did is systematically occlude one part of the image and ask the question, which part of the image, when we occlude it, will degrade the accuracy of diagnostic test. And then we'll put a heat map on this part of the image. As you can see in panel A, those heat map represents the lesion where occlusion map identified by the AI. This is precisely what physicians are looking at and make diagnostic decisions. So this is actually a very nice example how we actually look at the image and be able to tell and also help physician to tell where the AI was looking to make those diagnoses. The second part of panel B and panel C, our AI is automatically AI is guiding the anti-VEGF therapy for macular degeneration and diabetic macular edema, in which it identifies the lesions before and after treatment, and then tell the physician whether it needs treatment in the next round, when even before the physician see the patients. So this is again one of the examples of how the AI can help assist physicians in making decisions and let physicians trust where the AI was making decisions. So we then, using this pre-trained knowledge, the common general knowledge of recognizing pictures, in addition, the knowledge gained by training with 200,000 OCT images in eye space, to transfer one more time to learn how to diagnose chest x-rays in pediatric pneumonia. As you can see from the top and bottom pictures, those are totally different images performing totally different medical tasks. Yet in this case, even with 5,000 chest x-ray pictures, the AI was able to differentiate bacterial pneumonia, viral pneumonia, or normal. Differentiating bacterial pneumonia from viral pneumonia are important because those actually entails two different type of approaches. The antibiotics have to be given, and it's emergency referment for bacterial pneumonia for a pediatric pneumonia, whereas a viral pneumonia only requires supportive care. Our second case of AI assistance and AI physician interactions and trust entails using COVID-19 pneumonia as a test case. In this case, we were using 500,000 CT images to diagnose COVID-19 pneumonia in an early phase of pandemic, in which at the time the PCR or standard molecular test was not available, and physicians was swamped with looking at the CT images and trying to diagnose COVID-19 pneumonia. I shall mention that at the time, because most of the physicians in the world are encounter COVID-19 pneumonia at the first time, so they were just like novice or junior physicians are still start to learn from the scratch of this pneumonia. In this case, we were able to label the lesions, make a segmentation, and indicate the lesion subtypes such as ground gas appearance or consolidation fibrosis. In addition, also made end-to-end predictions for the diagnosis of COVID-19 pneumonia as well as severity of the pneumonia. The CT images diagnostic system performed very well in this case. In addition, as shown on the right side, the AI was able to give assistance or guidance or hint to the junior radiologists such that it can actually achieve a very high accuracy for diagnosing COVID-19 pneumonia or other pneumonias in very short of time. And showing in the lower right panel, the green ones are the AI diagnostic weighted error, and the blue ones in the middle are senior radiologists with more than 10-year experience. And on the right side are the junior radiologists for which they had made some significant initial errors and for which can be brought down very quickly by assistance of the AI network. Next, we will be able to using the lung function images as well as the clinical parameters to predict upon admission who's going to progress to the critical illness. This is crucial because those are the people who should obtain the most resource attention from the medical community, and the AI was able to predict quite well those people. So this is another example of AI helping the physicians and hospitals making very important decisions. And this system was able to diagnose COVID-19 pneumonia within 20 seconds. It's been deployed in many countries around the world, including China, and the second wave of the COVID-19 outbreaks in Europe and in other countries. And it was able to be uploaded to the China National Bioinformation Center with codes and data sets all available, shared worldwide, and so far has been downloaded over 3,300,000 times. Made its meaningful contribution during the pandemic, comeback pandemic. In order to also helping the other underdeveloped countries to diagnose COVID-19, we also used 150,000 chest X-rays to make another triage system. The benefits actually using as a frontline triage system to diagnose pneumonia and quick interventions or isolations for those patients for quarantine. I next want to talk about AI assistance and the empowerment using another AI modality, which is natural language processing and deep learning in diagnosing pediatric diseases. In this case, we're applying big data and AI for electromedical records, which is totally different from the images, to mimic and assist the human physician in decision-making process. In this case, we made a multi-modal AI diagnostic system using natural language processing and deep learning. I shall say that the electromedical records, the vast majority of them are going to be textual informations. In this case, we first use medical knowledge, guidelines, and consensus, and 50,000 medical record cases to make a question answer pairs. From there to deduce logic relationships and use natural language processing and deep learning to create a algorithm. This algorithm will apply to 1.3 million raw electromedical records, and to extract the information, form a fully structured data sets. From there, we undergo disease classifications. The AI can classify pediatric disease using a decision tree according to an organ system, just like human being in this case. And the AI-based EHR system can accurately classify pediatric disease and systems such as this. Finally, the AI's diagnostic performance is comparable to a mid-level physician's. The benefits is actually it can diagnose many rare and life-threatening conditions very quickly within minutes. In this case, we had a AI and a physician comparison in task performance. As you can see, the AI can perform quite well in all categories of these 5,000 different cases, and comparable to a level mid to senior level physicians. I also want to mention that, particularly in emergency situations, that not all physicians can be at its top performance level and cannot be able to diagnose all the conditions without errors. So this is actually particularly useful, I think, in the situations where the sepsis or important rare diseases, such as sudden heart arrhythmias conditions, can be diagnosed timely and quickly to address and to help with junior physicians on call. So at the time our paper was published, New York Times made a comments. I think it's actually illustrate this point very well. I just quickly read some of the comments. I quote, "Each time millions of Americans walk out of the doctor's office with a misdiagnosis, physicians trying to be systematic when identifying diseases and illness, but bias creep in, alternatives are overlooked." So this is precisely we're hoping that AI system can, at those situations, assisting our physicians, especially the junior physicians in a particular situation, in a rural area situation or resource poor area or on call. My last part of the talk is addressing AI empowerment in creating new knowledge and diagnostic paradigms. And you have heard from the last section that the AI now is poised to make a major impact in drug discovery and real-world clinical trials, and so to greatly accelerate the discovery and the implementation of innovative drugs and reduce the side effect and timeline. In this case, I'm going to talk about learning from random images to diagnose systemic diseases. We're all very well familiar that the AI can diagnose multiple retinal diseases, such as diabetic retinopathy or macular degeneration. I show you in my first part of the talk. However, it is to my surprise, I was surprised to learn that the AI was able to look at seemingly normal fundus images and making some very, very specific and highly accurate diagnosis of systemic diseases. So in this case, we were able to use AI system to detect the incidence, detect the chronic renal disease and type 2 diabetes, and predict the incidence from normal looking retinal photographs. In this situation, we used about 57,000 patients, 115,000 fundus photos, and combined with the metadata, was able to predict the chronic renal disease and type 2 diabetes and its incidence. Obviously, the benefits are with one single system, we were able to not only detect diabetic retinopathy diseases, we can now detect other diseases which is not actually possible by human eyes, not even by trained retinal specialists. The AI performance in identifying renal disease are very, very high, with the AUCs in the 90s. In addition, the AI was able to, we were surprised by these results, and we want to find out why, some partial explanations why actually the fundus photos can be used to do this. Therefore, we're looking for some of the parameters to explain this. Obviously, the first thing we look is, can AI, or was AI looking at some specific parameters that can predict? So we took the one single most important factors in renal function, which is estimated glomerular filtration rate, eGFR for short, and asked whether a retinal photograph can predict the value or measurement of eGFR. The answer is yes, it can predict eGFR with very high accuracy, as shown in here. Similarly, the AI was able to detect type 2 diabetes and measure fast glucose level. This is actually shown here, and in the panel A and also panel D. And different panels are either internal validation or 2 external validation cohorts. What surprised me the most is that using longitudinal cohorts with the baseline photographs, which is completely normal and does not have diabetes, we were able to predict the incidence of diabetes in the near future. To me, that's actually quite surprising, and then I hope this type of surprises will be actually more in the future. This is actually not only tell me that the retina contains information that's not actually discernible by human eye, but is discernible by AI that can be predicting some of the future events. And this is, I think, is actually a great way or example of how we can utilizing AI for doing some of the tasks which is out of the human recognition domain. Similarly, we actually tried the same approach where the AI was able to predict the incidence of glaucoma from normal baseline photographs in a longitudinal cohort. In addition, the AI was able to predict glaucoma progression from baseline photographs. Those are the early or intermediate level glaucomas. And then we're asking within next few years, which one of them will predict to be in a severe or end stage glaucoma? And the answer is yes, we were able to do both. So in summary, I mentioned to you some of the major challenges and opportunities in applying AI in medicine. And those challenges are data challenges and technology challenges and human resources challenges. I give you some examples of AI assistance in this process, in which we made a foundation or pre-trained models in transfer learning to make generalizable computer vision models to predict and diagnose imaging-based diseases. I also showed you the examples of how we can use AI to empower, to identify new paradigm and also new knowledge in the case of using normal looking fundus pictures to predict chronic renal disease and type 2 diabetes and glaucoma progression and incidence. Lastly, I talk about physician interactions for which the AI black box can be addressed by occlusion tests and also measurements of key clinical factors such as eGFR. I want to thank you for your attention. I'm happy to answer some questions. Thank you.
So our next speaker is Sherri Rose. She is an associate professor of health policy of Stanford University, and her research is centered on developing and integrating innovative statistical machine learning approaches to improve human health and health equity. And today, Dr. Rose is going to present toward minimum standards for healthcare AI. So, Dr. Rose.
Hello, my name is Sherri Rose, and I'm delighted to be presenting my talk on minimum standards for healthcare AI, with an emphasis on fairness considerations. We have many existing and new algorithms in healthcare, advances that hold promise to improve human health and health equity. I define algorithm quite generally as any rule that takes data as an input to return summary outputs. I'm going to start by orienting us on the current state of algorithms in our society. Algorithms are broadly pervasive and can cause harm. For example, Amazon wanted to automate their hiring. They created an algorithm trained on 10 years of hiring data for their technical roles. Unfortunately, because most hires had been men, the algorithm penalized the word women and all women's colleges. They tried to fix the algorithm, but ended up abandoning it. In another example, soap dispensers were unable to identify dark skin. The optic sensor to detect hands wasn't designed for darker skin tones. We see similar issues in facial recognition that can lead to dire consequences, where multiple Black men have been arrested in the United States for crimes they did not commit due to faulty facial recognition. Algorithms are also pervasive in healthcare. The data we have in healthcare includes information about health status, procedures, lab tests, medication, etc. And we'll be discussing issues with these data in a few minutes. Algorithms can range in complexity from a checklist in the emergency room that asks, should we admit this patient, to standard regression analyses aiming to predict length of stay in the hospital, to complex machine learning procedures detecting health conditions using images. The goal is to yield clinically or policy-relevant results to inform decision-making, spanning diagnosis, screening, prediction, and beyond. However, just as algorithms broadly in society can have a negative impact, especially for marginalized groups, this also happens in healthcare. Groups marginalized by the healthcare system include racial and ethnic minorities, disabled individuals, rural communities, people with lower socioeconomic status, and the uninsured, among others. These groups already experience lower life expectancy and poorer health outcomes. Algorithms could more frequently be contributing to improving health equity rather than harming it. We've seen racial bias in an algorithm such that Black patients would not get the care management they needed. There's also an opioid risk algorithm that surveils all individuals in the United States who pick up prescriptions, and it can lead to the denial of adequate treatment to those who need it most. So we know algorithms are pervasive and can cause harms, but how are we combating this issue? There have been various guidelines proposed, including for randomized trials and diagnostic accuracy studies. The FDA in the United States also has guidance for the clinical evaluation of software as a medical device. My own work has included several proposed standards and guidelines. These are just some recent examples, but uptake has been weak with no established minimum standards for general algorithms. This is a problem because it will not be feasible to study most algorithms in randomized trials. Additionally, existing proposed guidelines are insufficiently focused on ethics and fairness. This is discussed in the recent piece here by Gachoya et al. It's also important to highlight that ethics is woven throughout other areas of research, including conflict of interest reporting and IRB review, but not for the implications of AI tools. One of our recent papers included a proposal for an ethical pipeline for machine learning in healthcare, starting from problem selection all the way through to post-deployment considerations. This includes which topics are funded, how are research teams constructed, are you using non-representative convenience samples, do you define your algorithms using a variable that is easy to observe in the data, but may have differential mismeasurement, and what happens to the algorithm after it is implemented. I'll be revisiting the themes in this pipeline throughout my talk. To give just one prior example of the impact that specific requirements can have on the robustness of research, this plot displays results from a study of the volume of null results in randomized controlled trials funded by the National Heart, Lung, and Blood Institute in the United States from 1970 to 2012. You'll see that notably in the year 2000, pre-registration of primary outcomes became required on the website clinicaltrials.gov. Prior to this requirement, they found that 57% of trials demonstrated a significant intervention benefit compared to only 8% of trials after the requirement to pre-register their primary outcomes. That's 57% versus 8%. This is one compelling result among many regarding the impact of requirements established by entities with power in the research ecosystem. I'll next offer some perspectives on ethics and fairness in AI for healthcare research, although these will not be comprehensive. We'll keep in mind that the goal is to focus on avoiding the perpetuation of harm to marginalized groups, and I'll share ideas about reducing these harms. There are many questions we ask when thinking about fairness and ethics in AI research for healthcare. These are just a few and include: who has the power to decide the research question? Who's in the target population? What do the data reflect? We know that our data reflect societal biases, including structural racism and access to care in rural, minority, and low-income communities. We'll discuss this in more detail in the next few slides. Then, how will the algorithm be assessed? Are you only considering evaluation metrics for the overall population or also marginalized groups? We'll also come back to this later. A key tenet for fairness and ethics in healthcare AI research is centering justice, such that benefits, risks, costs, and resources are equitably distributed. And we know that the current equilibrium in our healthcare systems do not do this. Next, I'll discuss data and the evaluation of algorithms briefly in a bit more detail, starting with data. We know that societal bias enters both data and algorithms in many ways. Detecting bias can also be challenging, especially when we don't prioritize collecting the right data. On this slide, I have an illustration of some of the constructs embedded in the data that we analyze. We have the world as it should be and could be under a more just equilibrium. But what we actually have is the world as it exists, including societal bias. And what we analyze in our AI studies are data that are rife with many additional issues like non-representative sampling and measurement error. This can be especially problematic when these errors are greater for minoritized groups. To be concrete about healthcare AI applications, there are many different types of electronic health information that may be used to answer targeted research questions. I have several classes of data sources on this slide, but it does not include all types. The appropriateness and usefulness of these data sources will vary depending on the research area. We may also need to apply complex or novel statistical machine learning and AI tools that have desirable properties while remaining computationally feasible. However, many times the AI will be secondary to the importance of understanding the processes that generated the data, including the role of social determinants of health, as well as the limitations of each data source. For examples of these limitations, we know that healthcare billing claims here in the top left corner have very large sample sizes, but they lack much of the clinical information we may need to study something like the comparative effectiveness of treatments. We have newer data sources presented here in the darker blue color, such as imaging, wearable technology, digital information, and electronically aggregated news media data. An accelerometer like a Fitbit might appear better than a survey that has recall bias because the Fitbit collects such granular information on physical activity. However, these devices can suffer from measurement error and be good at measuring steps, but bad at measuring distance. The people included in wearable technology studies are also often not broadly representative of the target populations. Digital data sources like Google search trends and social media posts are often dismissed as being too noisy to conduct reliable studies. This overlooks that these digital data sources may contain information not available elsewhere, especially in regions without high-quality health data. Even in regions with high-quality health data, we most typically only have information for individuals who have an encounter with the healthcare system. This excludes many people, especially marginalized groups, from being included in studies focused on clinical records or billing claims, for example. The last area I'll discuss before a closing section of final thoughts is evaluation, including the impact of evaluation on generalizability. How we design and evaluate studies has a major impact on the generalizability of the research where we want to extend our findings beyond a study sample. This has implications for many different kinds of research questions, spanning prediction, clustering, causal inference, and beyond. However, generalizability receives the most attention for questions of inference. We may have a randomized trial or an observational study that we wish to generalize to a target population. We may also be interested in transporting these results to another target population. That said, generalizability is incredibly important for prediction or classification and clustering questions as well, despite the lack of attention these studies receive with respect to generalizability. In the healthcare and medical literature, machine learning and AI are predominantly used in these types of studies. Unfortunately, basic standards for published machine learning and AI studies for prediction and classification are lackluster, and these studies are unlikely to generalize or be broadly applicable for the improvement of health and health equity. Many of these studies are conducted with a single year of data in a single hospital or medical center. There are often no external samples or even cross-validation to partially address overfitting. There's also a focus on a limited set of evaluation metrics, with some papers only reporting AUC or R-squared. And the so-called AUC leaderboard is a popular way to evaluate tools, to decide which classification algorithm is the best, with a single overall metric that does not capture the totality of the performance of the algorithm. Algorithms can have reasonable or excellent measures of AUC and other metrics like false positive rate or positive predictive value, while having near zero measures of true positives. I have numbers displayed here that are not even the most extreme examples that we discovered in our recent paper on this topic. We also saw AUC values in the 90s paired with single-digit true positive rates. This is not only an issue for rare outcomes or extreme class imbalance for the outcome. We also saw these issues when class imbalance for the outcome was 30 or 40%, meaning that these metrics can be misleading when the outcome is observed in 30 or 40% of the study sample. A basic standard should be that we need a broad set of overall classification metrics reported that include measures of both discrimination and calibration. Additionally, we need to go well beyond this and center metrics focused on group fairness, where we also calculate metrics for minoritized groups. Common measures of fairness are based on this notion of group fairness, striving for similarity in predicted outcomes or errors for groups. If this large maroon rectangle is our study sample, and the individuals in our yellow rectangle are part of the marginalized group, we can calculate these group fairness metrics on all individuals in the yellow rectangle. We can then calculate metrics on the complement group, everyone in the maroon rectangle, but outside the yellow one. These are the individuals who are not part of the marginalized group. Fairness measures help us assess differential performance for marginalized groups. Will this AI tool perform worse for these groups? There are also new algorithms that actually prioritize these metrics jointly with overall fit criteria directly in the optimization problem. In closing, there is currently a proliferation of low-quality AI and machine learning research in the healthcare field. These papers include fundamental, basic issues that have major implications for our trust in the level of evidence that AI provides. This includes the lack of external samples, no cross-validation, and not considering multiple metrics, including group fairness metrics for marginalized groups. Overall, the research does not emphasize or prioritize algorithmic fairness and ethics for AI tools. The field would benefit tremendously from having standards drafted and adopted by the community where a broad and diverse set of perspectives are incorporated. This would need to be followed by buy-in and enforcement from journals and granting agencies. We know this makes a difference. Having guidelines that are not just suggestions, having guidelines that are just suggestions does not lead to change. It does not lead to improved rigor, robustness, or fairness. This may involve additional review criteria and processes for ethics. This idea has been proposed and implemented elsewhere, including at some computer science conferences, albeit without the minimum evaluation standards. It could also involve new ethics-centered board members at journals, staff at granting agencies, and ethics reviewers at both. What we need this to lead to is a minimum standard to require ethics, fairness, and health equity considerations for the AI tools. That a tool is robust only if it has been assessed for influencing or exacerbating harms to minoritized groups. Another path is adapting principles from previously proposed social impact statements and working towards specialized implementations that would be necessary for healthcare and medicine. Have the researchers established who is responsible to users if they are harmed by the algorithm? Is the algorithm explainable to users? How accurate is the algorithm? Where again, we would want many measures of accuracy. Is the algorithm auditable by a third party? Both the algorithm itself as well as the data. This is where we may need especially creative solutions in healthcare and medicine, given that we work with highly sensitive data. Solutions could involve external auditors being added to data use agreements and IRBs. The status quo is very far from this. We have many AI publications in healthcare that do not include any real description in the manuscript or appendix of the underlying architecture for the algorithm, and certainly no open source code. Lastly, we discussed some aspects of fairness. Have the researchers considered the fairness implications of their tools? The potential benefits of such minimum standards are enormous. This can result in improved generalizability and transportability to diverse populations. It would lead to attenuated claims in the literature regarding readiness for translation into practice, as well as better contributing to improving health equity. I'll end by saying I am incredibly fortunate to work on these issues with a wonderful multidisciplinary team of trainees with expertise in informatics, medicine, computer science, health policy, and economics. I thank them for their many contributions to our research. I also want to acknowledge my funding that helps me advance this kind of work. Thank you very much, everyone, and thank you for the invitation to present these ideas at this event.
Thank you so much, Dr. Rose, for this very thought-provoking presentation. So next, we're going to move on to panel discussion. So in addition to the two speakers we just heard, we have 4 more panelists to join us. And let me introduce briefly. So we have Professor Enrico Correia from Macquarie University in Australia. So He has a strong international research reputation for his work on decision support, digital health safety, and communication processes in biomedicine. And we have Dr. Qingmei Gao. So she is a professor in Tongji Medical College at Huazhong University of Science and Technology from Wuhan, China. She excels in the surgical and systemic treatment of gynecologic cancers and dedicates to the precision and translational medicine of ovarian cancer. And so Dr. Tiannan Guo is from Westlake University, and he is trained as a clinician, but his research focuses on proteomic technology development and AI-empowered applications in disease. And so this panel will be moderated by Dr. Zach Cohen, and he is an inaugural chair and endowed professor at Department of Biomedical Informatics at Harvard Medical School. And he's also an editorial board member of New England Journal of Medicine. So Zach develops and applies computational techniques to address diseases at multiple scales, from whole healthcare system as living laboratories to the functional genomics neural development. So without further ado, so let's give it to Zach for the moderation of this panel.
Thank you, John Fay, for doing a terrific job in running this meeting so far. Thank you all for the invitation. Thank you to our panelists. And so I'm going to, we really only have 40 minutes. We're going to have to end at the half hour. And so we've heard so far in this session a tremendous presentation on both how AI can be used to perform at the level of the most competent doctors, and sometimes even in superhuman ways, such as looking at signs of renal disease by looking at blood vessels and other features in the eye, in the retina. We've also just heard a sobering review of the limitations of these techniques and how we don't have a lot of transparency and insight into essentially what assumptions are baked into the techniques and where these techniques may be brittle or not. And I am reminded, and so I have to say, speaking personally, I'm very optimistic about this field. And I think that we'll be hearing more over the coming years about the impact that AI will have for good in medicine. Nonetheless, I think we're really in the, speaking as a pediatric endocrinologist, we're in the era of growing pains. I am mindful that there was a well-intentioned multi-hospital predictor of morbidity and mortality developed from the EPIC electronic health record at several hospitals that was built in the anticipation that we might have to triage patients. And this predictor was shown by other hospitals using the same electronic health record system to be extremely brittle and not be reproducible at sites, at different sites than where it had been trained. And moreover, because both the biology of the disease changed given the vaccination status of the population and due to the rapid change, thankfully, of medicine to use techniques that we found to be effective, like the use of dexamethasone and other glucocorticoids, that even the original hospital where it was trained up switched off this predictor because it was no longer valid. And I don't think anything happened here that surprised anybody, didn't want anybody to happen. But we're about to be faced with a profusion of our AI artifacts. And we haven't yet heard from three of our panelists, but whether looking at Dr. Go looking at proteomics, or Dr. Gao looking at various oncological markers, where there's going to be a profusion of such markers. How are we going to know when this is safe to use with patients? And in order to get the conversation going, I'm going to ask each of you to be brief and say, what is the one thing? The one thing. Well, then we'll have a conversation, especially if we're brief. What is the one thing that you think we could do that would give you and doctors the most confidence in using AI for clinical decision-making, for biomarkers, for all the kinds of things that we just described? And just because I like to pick on friends, I will pick, first of all, on Professor Koira from Macquarie. So, Enrico, what is the one thing we could do that would give you and the rest of us more confidence that these algorithms are safe and or fair?
Thanks, Zach. Can I cheat and say everything that Sherri said? Plus, I would like to add in, the importance of replication. And what do I mean? We have to trust the evidence base. We have to trust the research. And the way scientists do that is to independently validate the work of others. And we don't do that in this discipline. And when I say we don't do that, I mean that because I undertook a study just a year or two ago where we looked at 4,000 papers, clinical use of decision support. We found 12 replication papers. 0.3% of the literature on clinical decision support was any form of replication. When psychology declared they had a research crisis, they had a replication rate of 1%. So we're 3 times worse. So a lot of the things that Sherri mentioned around changing culture and best practice extends to recognizing that just building and publishing new algorithms is not science. Science is carefully evaluating progress. It's a change in our attitude to what we value as researchers.
So Enrico, let me put you on the hot seat here. So someone just publishes a 500 gigabyte foundation model, and based on this, they're showing great prediction of kidney disease based on just electronic health record data. How are we going to independently replicate this? Are they going to give me their 500 gigabyte foundation model? How are we going to do this?
Yeah, they don't have to, Zach. There are different layers of replication, and you could say, I need to have all your code and all your data, but you could put your algorithm behind a firewall and allow me to use it independently with my patients or in my clinical setting or in my country to see if I come up with the same answers or not. So the idea that, the great is the enemy of the good here. There are ways of doing it. And, Sherri talked about, yeah.
That was a not a rhetorical question. I wanted a constructive answer, which you give. Dr. Gould from Westlake, you're involved in a variety of very important biomarkers. How are you going to, and I do remember back when we had gene expression biomarkers for breast cancer, there was some replication issues. And so how are we going to trust these new biomarkers that you're going to come up and give us that we're going to have great performance? What is the one thing we could do that would give you if one of your colleagues published something?
Yeah, I think we need a more systematic measurement of molecules in the clinical specimens. So just now, Dr. Zhang and many others mentioned text data, image data, they play a very powerful way in improving the AI diagnosis and treatment, but they are not touching the essence of the disease. For the essence of disease is not only genes, not only transcripts, but other molecules like proteins, metabolites, which cause the disease and which are the therapeutic target of drugs. So I want to emphasize systematic measurement of the molecules in clinical specimens. Thank you.
So systematic measurements of these molecules, so to allow comparability, for example. And do you think we are? How close do you think we are to that? I shouldn't. That's a rhetorical question. My impression is we're not that close to it. We're barely able to reproduce whole genome sequences, and I worry that in your area of proteomics, how do I know that someone's mass spec pipeline is similar to someone else's mass spec? They may be using the same machine and just slightly different settings and different prep, and they can have quite different results. So how do we get around that?
Yeah, there are several issues. So I think it's not only about replication, but also to eliminate the data bias. I want to say that the text data, image data, they are actually a bias for AI-based application, because we are looking at the morphology, looking at the patient's complication. They are not the essence of the disease. But for the second issue, how reliable is this molecular measurement? This has been a long under debate question, but I think many people think this is the technology for the future, particularly for proteomics, because they are so expensive. They have to use a big piece of tissue which has been properly stored and prepared, and they are so sophisticated pipelines. But this has been changed during the past few years and keep changing in a very rapid pace. So today, I would like to say, if you have a needle biopsy, which is really very small, you can hardly see by your eye. If you have a section of an FFP slice, and we can robustly measure the proteins, not only hundreds or thousands, up to 10,000 proteins, reaching the depth of a transcriptome, maybe at a reasonable cost. It's not as cheap as RNA-seq, but the cost is reducing very rapidly. So I think I'm very honored to participate in the meeting last year, and this year we get even better progresses. And I believe in another 1 or 2 years, then a lot of clinical specimens can be have a protein profiles and also other molecular profiles at reasonable cost, and they may play a much better role in AI-based medical science.
Professor Gao from Tongji Hospital, would you opine upon what is the one thing you would do that would give you confidence when you or your colleague, where one of your colleagues or someone that you didn't know published an AI study? What would give you confidence?
Okay. Yes, I learned a lot from here. Because I'm a clinician, I'm a surgeon and focused on organ cancer, no matter research and also the treatment of the patients. So AI helped me to diagnose the image from the ultrasound image. So we constructed the model, and with 600,000 of images from 120,000 patients. Yeah, it's a lot. I just learned a lot, and I trust with this AI model, we can help patients for the diagnosis for differential of the organ benign mass from organ cancer. That's very, very important for the clinician. So I'm a new younger in this field. So I think this study,
So let me ask you, why do you trust this huge data-based ultrasound modality more? Would you trust it more than a seasoned ultrasonographer who you've worked with her, she's fantastic, and you really trust her? What makes you trust that they had all the right cases in the database of the ultrasound? They're not going to miss some weird cancer or some weird anatomy which fools the ultrasound. Why do you trust that AI characterization as much as you would trust the characterization from your ultrasonographer colleague? Or is it that you don't have such a trusted ultrasonographer colleague?
Yeah, it's a very important question. Yes, the first I use this. I collaborate with my the professors in the AI field. We use. I think it's amazing. First, amazing is that when it's very, very difficult to diagnose use ultrasound image, but with this big data information, image, I think the AUC is higher in the almost 90%. Yeah, it's very, very amazing. So then it let me to collect more and more images, and with the more images, I think it's a more compatibility. So because it can combine the vaginal images and also the abdominal images. All the images we can confirm it's a malignant tumor and not a benign tumor. Especially from it's most impressed me most is we found the 12 patients with a small mass in the ovary, and we dynamic monitor its changes, and with the AI we diagnose it before the surgeon, before the clinical detection, almost 2 years. It's a very impressed me most. I think so. I trust it. We use it to diagnose the ovarian cancer. I think more faster, more accuracy. I think it help us a lot. Yes.
So it seems to me that what's convincing to you is actually seeing examples where there are potential lesions that might have been missed or found early in the process, and seeing that these are detected yourselves is what is convincing to you. So thank you for that. We'll get back around to you, but I want to make sure we do a first round of questions. Professor Zhang, like I said, you showed some very impressive work. What if there was another Professor Zhang out there who was showing equally impressive work? What would be the one thing that gave you the most confidence that the samples from which this was drawn was sufficiently representative, and that the accuracy was such that if, God forbid, a loved one of ours would have this problem, we would be comfortable using it with them?
Thank you, Zach. And I think it's, I actually, first of all, echo all the speakers and panelists' actual opinions, and particularly Yuriko, that replication, and generalizability. And there are many ways to do that. I think, you mentioned some of the using EHR to predict, for example, serious events, outcomes, or death for that matter. And because we have lots of EMR data, and it's just a matter of how to actually preserving privacy and then equity, and just such as the Sherri mentioned in her talk. And with that as a precondition, I think it's just using the data to replicate and real world data to replicate in different settings, in different populations, in different countries will be very, very important. And there are ways to actually to do this, even with privacy preserving, privacy preserving matters such as federated learning and some of the swarm actually technology. And then lastly, I think one of the other things is actually just you give it to the patients, have them actually in their end, and then without just install, for example, in the iPhone or in their computer, and then without data actually coming out of them, and to monitor them in a massive way and the most normal way as we will use them, just like an iWatch or iPhone, in a way to monitor the condition or diagnosing conditions. That's first. This is actually, as long as we have some data with a different new old modality, that's I think it's a easier issue to address. The question is actually if someone invented, Dr. Guo invented a new algorithm, and then it's brand new, and we have to test this somehow, and then there comes to just how to assess the code and data, and then to have them basically popularized and then or being tested. This is a process, and we kind of have to do this. We have always been a staunch supporter and a believer for sharing data and codes, and all of our actually published papers in what you see are actually the codes are all being shared, and also and the data has been most of the data apart from some of the data which require for privacy are being actually published. So this is why actually our papers have such a high citation rate, because people will use the data to do further analysis. Thank you.
That's that great answer. Professor Rose, you touched on so many different things. So I'm going to be a little bit mean to you by asking you, of all those things that you mentioned, what is the one that's most burning? What would be the thing that you think could help us the most?
I think if I had to pick one that aligned with some of the other panelists, I would pick external validation. So a genuinely external sample, most optimally in a patient population that has a different composition than the one where the algorithm is trained. If we had that requirement, I think the majority of AI and machine learning papers wouldn't even be published, because so many of them don't have that. And I think it would really raise the bar and the quality and really help us understand where these tools work and don't work. And I really want to also specifically support the replication comments that came from a couple of the panelists, because that would be another culture shift for us to actually incentivize people to do replications, because right now the incentives are not there for folks to pursue those. And if we're actually trying to pursue, improving human health rather than careerism, then we should be doing many more replications.
That's hard to argue with that. But now I'm going to start with you, Sherri, Professor Rose, and we're going to go now backwards through this crowd, because now that you've all articulated that one thing, I want to wave a magic wand. And that magic wand is creating a new, very, very impressive journal that we are all going to respect. What is that journal going to be able to reasonably ask of the authors, the investigators, to do that thing that you said was most important? What are they going to be reasonably asked? And as you pointed out, a lot of the current trials don't meet that criterion that you just set. So does this mean that this very important journal will have nothing to publish? What should it be asking? What is this journal that we're all going to respect going to be asking of its submitters?
Well, specifically for the, having an external validation sample, that one, and I'm not just saying this because New England Journal of Medicine is part of this event, but I haven't seen any AI or machine learning paper in the New England Journal of Medicine that doesn't have an external sample. So there's a journal where it's already de facto required. And so I don't think that this even necessarily has to be a hypothetical journal, but it could, the same way that journals have these requirements for the reporting of statistics. The New England Journal already has a requirement for diversity in research studies. So it would be in some ways a trivial additional requirement to ask that you test your algorithm in an external sample. So that one, I think, is it's really the totality. When we get into the issue of wanting to address all of the different things that have come up, including many of the topics in my talk, that it gets a little bit more challenging of how to actually operationalize that. But I think external sample is really feasible.
Well, that's incredibly helpful and clarifying, and you're absolutely right. That we do have some notable examples, including our colleagues here at the New England Journal. Professor Zhang, I'm going to ask you the same question now, which is, magic wand, what is this? Let's say you're going to have a new publication about and you're going to be able to predict Alzheimer's, looking at the back at the retina. What can this new journal expect from you in terms of providing convincing evidence?
Well, obviously, Zach, this is actually a very important question. And I think that just for a journal, actually, as an author and a reviewer, what I always believe in is actually those studies you need with a goal. The overriding goal is always going to be actually, does this actually change the paradigm? Does this actually influence clinical practice? Does this actually help us to solve questions that we was not able to solve, or serve our patients better? With that, was basically precondition. I think the generalizability and replication has to be very, very, very important. But this is actually this is going to be a bit of hard to address. Let me give you the reasons. First of all, the health data, especially the electron medical record data, and I'm assuming eventually DNA sequencing and some other retina or facial data is going to be tightly regulated. So with this background as a drop, and also different countries have different ways of regulating those data. I know actually China has very strict policy in actually data. So with this as a condition, how would we actually be able to replicate and implement those algorithms and serving our different populations, especially underserved populations, and for which I'm now based in China. It's very hard for me to get actually non-Asian ethnicity data. Rather than actually if I don't through collaborations, right? And so this is one issue. I talk about some of the other issues using federated learning or edge computing to solve those issues, and this is also better in a particularly unfortunate time of geopolitical instability. So it's become harder and harder for scientists from different parts of country or world to collaborate with each other on those type of projects. So those are issues. I don't know. I've been actually thinking about how to actually creating synthetic data and using some of the data that is actually does not belong or actually have some generalizable features that I'll be able to use as a replication data. Obviously, if all of us actually work together, creating some kind of the repository data sets that can be used as benchmark for some of the studies, that'll be wonderful as well. Thank you.
Thank you. That's extremely thoughtful. And I think you give us an important reminder that even well-intentioned well-intentioned protections of privacy and of the individual can end up having relatively unexpected and untoward effects. And so it's not just China, but Europe, for example, has also extremely rigorous standards about sharing data, like genomic data. And of course, why should we can only expect to increase the use of genomic data alongside the more conventional clinical data. And you're quite right to point out that the current regulations provide a pretty serious activation energy to overcome, and that slows down research at the very least. And with regard to synthetic data, I have to say my own personal bias is that whether you use differential privacy or a variety of aggregate techniques, often in order to be privacy preserving, you actually end up erasing the particular characteristics that you want to preserve in the local data sets that would address some of the issues of representation and equity that Professor Rose raised. So I'm glad you raised that issue, Professor Zhang, because I think it's an unpleasant one, but it's also, I think, something that we need to figure out how to deal with. Dr. Gao, it's great to have a young clinician working here, and I'm sure many of us wish we had that perspective. But given nonetheless, if you, what would you expect of a journal that was publishing perhaps a study on detecting small lesions in ovaries for women over 60? What should the journal ask for in doing this study that would give you confidence?
I'm sorry. Can you?
Let me try to rephrase that question. You gave a great example before of how a machine learning approach to the ultrasound identified early small lesions that allowed for early detection. If these were published in a medical journal, what would give you the most confidence? What should the journal ask for? Should the journal say, oh, it's okay to just use the data sets that you trained up with? Or should it say you have to go to a different provenance in China that was never used? Or even it has to be tested in people outside China? What should the journal be asking for in terms of making sure that you can trust the findings in a publication?
It's a very good question. Yes, I think if we publish the image with AI help the model, we should at least validation, at least in the internal validation, and it's better to use it in the external validation. We publish our work in the Lancet Digital Health. I have already introduced it with one internal validation and two external validation. I think it's at least it should be validation, and if it's better, I think if we can help a lot with this, if we can show evidence this model is worked okay in the practice, it's better. So we use this model help our radiologists to use in their practice. So that is, I think it's better because it could be help our younger radiologists improve faster and act with accuracy. I think so. That's why you ask me why I trust AI, because it helps us a lot in our clinical practice. I think I believe it. Yeah.
Great. So I'm now going to go to Professor Go, and you suggested already something about having these shared, standardized benchmark data sets. But given the fact that right now not everybody will have access to the same machines that you do or the same techniques, how do you go about validating your publications? What would you expect of a validation? Do you get specimens from elsewhere? Do you have what do you do in order to get the kind of validation that we just described in proteomics?
Yeah, thanks for the question. I think this is a very important issue in omics research, not only proteomics. So people need a standard, need the same or some sort of SOP for extract molecules from specimens and measure it with a certain level of QC and reproducibility and so on. But for proteomics, there are multiple standards. Means each one of them looks reasonable, but there's not only one standard applies to every proteomic field. And even worse for metabolomics. Probably for genomics, this is more standard. So that's why the NEJM and many other journals and many other authors apply the genomics approach in your research, and they showed very good results. So I've written some articles saying we need a standard. But I also want to echo to some of your previous question. You see what respected journal can do to promote this. So ideally, one paper can answer all the questions about replication, generalization. But in reality, due to the restrictions between different countries, many other issues that you all know, probably is not feasible. Or I think any respected journal and respected authors, we share the same goal to promote medical practice. So how we can do this? You can ask the authors to provide 2 independent or 10 independent validation before they can publish their paper, or we can do something more constructive. Constructive. For example, I don't know whether it's possible for the journal to coordinate the publication of several specific fields. For example, the imaging, for example, against COVID or another pneumonia. You know better who is doing what in a certain time. And then you can publish in a special issue so that different group, they can validate the data from each other, or you can help to coordinate them into consortium, international consortium. And another point I want to make is, you emphasize the usefulness of tools. I perfectly agree with that. But for a new tool to penetrate into clinic, this has been extremely challenging because the new tool is immature. They have limitations, although they have a promise. So I'm not sure whether a respective journal can do anything to improve the penetration of new technology, new tools, like proteomics. So I think everyone agrees the importance of protein measurement levels in disease and in drug discovery. But till now, there's no protein-based measurement. Proteomic-based measurement has been really applied to clinic. So we have been trying very hard, but there are a lot of requirements from many respected journals, and this has been even more challenging for new technologies. So this is my some suggestions. Thank you.
Thank you. Very thoughtful. So Enrico, you get to close, and I'm going to make it difficult for you. I've talked to some well-meaning and thoughtful regulators who are aware of the fact that populations are different in different locations and that the practice of medicine is different. So Dr. Gu of Westlake tells us difficulties he's having standardizing proteomics. Standardizing medical practices, a much taller order. Could we even request, would you even trust a replication that happened on a different EHR? And how stable would you think it is? Or, for example, would you think we should require some sort of pre-training or transfer learning to the local system and to show that somehow it's more robust to a data set shift? Just think out loud, given electronic health record applications, using the actual output of humans dealing with their patients and all the geographic and cultural differences, what looks like a solid replication that you trust?
Thanks, Zach. Just to summarize what you've said, the question is, can we or should we even try to replicate when context is such a confounder in evaluation? And the answer is absolutely yes, we should. And the answer is that we already do it in healthcare, in health services research. Context is a standard problem. We have something called implementation science. There's a whole discipline devoted to understanding how to solve these questions. If you look at the replication paper I mentioned from 2021 that we wrote in JAMIA, we actually show that it's important to measure difference because sometimes a replication tests the truth. Did your experiment work? Did you make a mistake? Sometimes your experiment tests the generalization to another population. And the fact that I couldn't generalize to my population with a different disease doesn't mean you failed. It just means it doesn't generalize. So I think if I can sneak in an answer to your question, what do we need to do? I would love to see a journal of replication in health and biomedical informatics. That would change the culture so that we start to value it. It would give us a venue to publish because the truth is big journals like New England value novelty. You love a new result. You're not going to publish a replication that shows those guys were right. But that's important science that needs to be done. So let's have a journal there. And that question you started with around context and the importance of variation in outcome is a basic implementation science question, which we need to develop new methods for. So let's put that into that journal as well. And Zach, you could be the first editor.
Wonderful. Thank you for that yet additional monkey on my back. I guess I deserve it. Thank you all for being so wonderful and thoughtful and really insightful. I think you really articulated some of the big issues that the policymakers and scientists are going to have to address. I am saddened, but I think important that we brought up the international one with the changes in with the different privacy laws, because I think that we need to talk to our leaders about how to address this because it's going to affect their care. And I have to apologize now because we've run out of time. And I would like to hand back the baton to John Fei, who's done such a great job. And thank you all for participating. John Fei.
Thank you, Dak. So we actually covered many important and thorny issues in medical AI in this session. And I think, this is not the end of discussion. This is actually the end of the beginning. Let's paraphrase some old sayings. But I hope that, we continue this conversation in the either, in AIMS in the future or, in the new forum, in the new journal launched by Zach, according to Enrico. So I don't know, hopefully. So anyway, it actually concludes our day one program. And we actually have some futuristic thoughts by our young and upcoming clinicians and AI scientists for our Day 2 program. So please tune in and let's see you tomorrow. Thank you.
Thank you.
Thank you.
Bye.
Great.
Bye.